Hello, my name is Richard Versaput with S-Matrix Corporation, and I'd like to welcome you to this presentation of LC method development for large molecules with Fusion QbD. This video presentation will use a recently completed project on development of an ion exchange method for monoclonal antibody charge variant analysis to illustrate the standard method development workflow in Fusion QbD. As we will show, the Fusion QbD tools, capabilities, and workflow presented in this video are completely aligned with the ICH Q14 and Q2R2 guidance documents, and also completely aligned with the USP 1220 and 1210 guidance documents. The Fusion QbD software platform has tools and capabilities to support all the critical elements of the analytical procedure lifecycle management workflow shown here. In this presentation, we will focus on the Stage 1 elements of robust method development, replication strategy optimization, and sample preparation method optimization. This slide shows the standard method development workflow in Fusion QbD. You execute this same general workflow for all your method development work. The steps in the green background indicate specific alternative settings depending on the experiment goal, the separation mode, and the sample type, small or large molecule. For example, in experiment design, you may select a separation mode specific to large molecule method development, such as ion exchange or size exclusion. You may also select very different metrics of peak shape and separation for large molecule method development. In this example, we will use the peak-to-valley ratio as a separation parameter as presented in USP 621, since in this example, the acidic and basic peaks on either side of the main peak cannot be baseline separated. We will also include the retention time delta, defined here as the retention time difference between a given peak and its immediately earlier eluting peak. We will model and set combined PV ratio and RTD goals for the critical peaks to achieve optimum mean performance and robustness in the final method. This slide presents the combined peak-to-valley ratio and retention time difference method development goals for the critical peaks. Achieving these goals will support the ultimate use precision and accuracy goals for quantitation specified in the analytical target profile. The excerpts from the ICH and USP guidances quoted here state that the design of experiments methodology is a proven and accepted way of quantitatively characterizing the independent and interaction effects of important method parameters on critical method performance results. We start our case study example with a very simple chemistry screening study, which includes 3 different paired buffer systems, each with different strong and weak mobile phase compositions, and each providing a built-in pH gradient. Gradient time, which is exactly a gradient slope factor given a constant starting point and end point strong solvent, and 3 different columns, each with the same dimensions but different stationary phases. Once the experiment is run and basic chromatogram processing is complete, we need to consider which results to import at the screening stage. In this example, we will investigate the number of visualized peaks in each chromatogram and also the peak-to-valley ratios of those peaks as an initial shape parameter. We will use the flexible and user-configurable trend response capability in the Fusion QbD import wizard to import 2 responses from each chromatogram: the number of peaks which are sufficiently separated to be integrated as unique peaks, and the number of the integrated peaks in each chromatogram for which the smaller of the 2 peak-to-valley ratios is greater than or equal to 1.2. Note that for this data we do not need to track and identify each peak in each chromatogram, which would be extremely difficult at this screening stage. With one click, Fusion QbD instantly models all imported results and can use the models to predict method performance throughout the study region, including excellent visualization graphics. To identify the best performing method overall, the user defines a minimum performance threshold for each response. The numerical search engine then uses the models to search through every possible method to identify the one which has the best overall result for all included responses simultaneously. The best overall answer is then presented in a report which includes the method settings and the associated predictions for all responses included in the search. The models and associated search results can then be visualized graphically. The 2D graph on the right side of this slide shows a nice feature which lets the user accept or adjust the defined minimum performance threshold for each response and add an associated color. In this graph, we have initially included just the number of peaks response with red as the associated color. As the graph shows, Fusion QbD has sliced through the response surface at that point to easily show the portions of the overall study region which contain methods with acceptable performance for the response, the unshaded regions in the graph, and the regions containing underperforming methods, the regions shaded in red. In this version of the 2D graph, we have included the number of peaks with peak-to-valley ratios greater than or equal to 1.2 as a second graphed response and assigned it the blue color. We now can see the portions of the overall study region which contain methods with acceptable performance for both responses simultaneously. In the follow-on optimization experiment, we include the best combination of buffer system and column type along with the operable range of gradient time. The optimization study also includes pump flow rate, column oven temperature, and initial percent strong solvent. In this study, the import wizard will automatically import all responses required for resolution modeling, generating prediction chromatograms, and for Monte Carlo robustness simulation. In the optimization experiment, we now include the mean performance goals specified in the ATP to see if one or more methods can be found which meet all performance goals simultaneously. As before, the best overall answer is presented in a report which again includes the method settings and the associated predictions for all responses included in the search. Here we can again view the location of the predicted best method within the joint study variable ranges, and also the size of the unshaded region around the method conditions. The statements in this slide present that integrating quantitative robustness into method development is aligned with the regulatory guidances. Therefore, once we have identified that one or more methods within our experimental study region can simultaneously meet all mean performance goals, we can then investigate whether the methods can also meet our method robustness goals. In the robustness simulator wizard, we can define both the expected total variation in our critical method study parameters on transfer and normal use over time in the QC environment, and also the method robustness goals for all included critical method performance characteristics. Here we see that the robustness performance requirements have automatically been added to the acceptable performance region graphics. In this case, they have all been assigned a gray color. And the updated graph shows that not all of the previously unshaded region designating acceptable mean performance is also unshaded for robustness. It is in this final unshaded region where methods are found which simultaneously meet mean performance requirements and robustness for all critical method performance characteristics. This slide shows a four-factor trellis graph series in which the previous 2D graph of responses relative to pump flow rate and gradient time has been replicated across user-defined low, medium, and high level setting combinations of initial percent strong solvent and column oven temperature. The graphs also contain the rectangle designating the transferable method operable design region, or motor, with red dots identifying the minimally sufficient set of motor verification runs. These runs can also be automatically exported to Empower to generate confirmation data and chromatograms. Once the final robust method has been determined, a replication strategy study can be carried out in Fusion QbD to identify the optimum combination of sample preparation and sample injection repeats to generate reportable values which meet the quantitation performance requirements of the final method. The replication strategy study identifies the relative contributions of sample preparation error and injection error to the total analytical error. It also identifies all strategies which meet the quantitation performance requirements of the final method, so that the user can identify the most efficient strategy in terms of time and cost. When the study does not yield an acceptable replication strategy, due for example to high sample preparation error, as shown here, then Fusion QbD can be used to optimize the sample preparation method in terms of the quality and repeatability of all critical performance characteristics. This slide illustrates the experiment setup for a sample preparation method optimization study within Fusion QbD, and also the four-factor robust method operable design region established from Fusion QbD modeling of the experiment results. It is noteworthy that the optimization was able to identify a robust final method which eliminated the sonication step, a major contributor to the variability of the original sample prep method. Once the sample preparation method has been optimized, the replication strategy study is repeated to identify the optimum strategy for generating reportable values which meet the quantitation performance requirements for the analytical method specified in the analytical target profile. The replication strategy then becomes part of the final method protocol transferred to QC for method validation. This slide summarizes the work and results presented here for the rapid development of this CIEX method. This slide summarizes the work and results done using the same method development workflow for another recent study which developed a robust HIC method for drug-to-antibody ratio analysis of antibody-drug conjugates. In conclusion, this presentation has shown that Fusion QbD fully supports the robust development of large molecule methods with a standard fully supported workflow which is completely aligned with the current regulatory guidances and expectations. That ends this presentation of LC method development for large molecules with Fusion QbD. Thank you very much for listening.